My name is Reid Olsen. I'm a third-year graduate student in the Department of Pharmacology at UNC Chapel Hill. I'm originally from Portland, Oregon. I went to the University of British Columbia for undergrad. If you think about our brains, it's really this food computer, but what's interesting about it is that it's able to reflect on itself. It's able to think about itself. So when we think about why we're here, if that's the kind of question you're interested in, or what we do, like what is the purpose or the ability for us to do things? Do we define our own meaning? Became very important to me. I wanted to pursue that. I wanted to understand myself. I wanted to understand people, why we're so different. How do we form these important relationships? Why do things matter to us? And how do we decide what matters? I had this incredible high school biology teacher who was just absolutely amazing. I remember struggling and one day everything clicking, and I just decided that this is what I want to do. And from then on, I just had amazing professors, amazing teachers, and not just that, but mentors in my life that I've sought out and have been really helpful. It's been a lot of personal ambition, but I can't tell you how important it is to collaborate and work together. What we study is how new neurons are generated in the adult brain. And one of the things that's very difficult about working in neurons is that there are so many of them. In fact, there are more different types of neurons than there are other cells in any other system. So to figure out how different systems, different parts of the brain, different chemicals contribute to the regulation of neurogenesis is very complicated. At UNC, we have access to a technology called DREADS. They're a really incredible, powerful tool that were invented by a collaborator of ours, Dr. Brian Roth. And what they are is essentially a synthetic receptor. It's called chemogenetics. It's genetically encoded. You can use like a virus to express it. And what's cool about these is that the receptor has been engineered in such a way that it doesn't respond to other neurotransmitters in the system. All the chemicals in your brain that are used to regulate neural activity don't do anything to this receptor. And it's absolutely very powerful. This is where we make a lot of our electrodes and optical fibers. So we've got these fun little lasers for shining lights to activate neurons. So if you express that in neurons and you use this green light to activate the ion channel, you can actually decrease the membrane potential in the sense that it becomes more negative. We can look at these waves of calcium in the cells. So this is a video we took of some cortical astrocytes. You can see we're going to add the drug. You can see here these cells, and you can see the kind of the change in the brightness as it kind of moves throughout the tissue. The field that we work in is very new and very poorly understood. So we're trying to answer some very fundamental questions here that until recently we haven't had the technology to really test. It seems like there's a lot of evidence that in depression, in epilepsy, in Alzheimer's, that neurogenesis is specifically affected. Maybe we can develop targeted therapies to treat those components. Collaboration is critical to science. We have these open labs and you can walk down the hall. I can walk into the office of almost any professor here and sit down and ask them questions, and there are no hesitation in providing resources and education, and they take it very seriously. And that's a big part of the ethos here. Having as many people involved, making science less isolated is critical. I can find something out and it's something that no one else knows. And while I want to share that knowledge, there is something uniquely amazing and joyous about being that first person. Science is accelerating. It's always increasing its speed, its discovery. And I hope that we do find some way to translate some of this, or at least understand it and push people forward. I think people look around and they see something immediate, but they don't realize that all of those things were dreams, were mysteries, were impossibilities in the past.